morning. I am from Team Specimens, and our problem statement focuses on identifying taxonomy and assessing biodiversity from the eDNA databases. The experience problem with the traditional eDNA pipelines is that the QIME2, DADA2, and Martha rely heavily on the reference databases. This makes them less effective in detecting novel or divergent deep-sea taxa. Moreover, their database dependency often limit accurate biodiversity assessment. To address this, we propose a 3-layer probabilistic model that provides a more reliable classification of marine phyla. The solution encompasses a complete automated pipeline that takes raw sequencing data, trims based upon quality distribution, and validates the completeness of sequencing process based upon location-specific gene segments. In the first layer, a model applies probability distribution over sequence features, example KMS. This layer consists of an unsupervised clustering model trained exclusively on marine data. It groups organisms into clusters corresponding to phyla and estimates the probability of that sequence belongs to an existing phylum or a potential new one. To validate this, we run test data against each phylum and measure the probability of correctly assigning existing sequence to their respective phyla. The second layer is a supervised model that uses proteome information from the phyla predicted in the first layer. This model determines whether a species is more likely to exist in intertidal zone or the deep sea, correlating the results with the probability distribution from the layer 1. The third layer also employs a supervised model but directly analyzes raw 18S DNA sequences. Here we apply the Chow-Fasman method to identify the loop regions in the RNA structure, as the loop RNA structure is more prone to mutations and often influence transcriptional protein binding. They provide useful signals for phylum level classification. This structural validation is exploited to refine prediction. Each of these layers contributes to weighted probability score. Additionally, we incorporate conventional BLAST but with two enhanced approaches. Firstly, running BLAST of 18S segments against the entire marine phylum database, and the next optimized BLAST, which is run exclusively on marine datasets, both rich and poor, to fine-tune the weighted scoring. Finally, our decision function integrates the result from all the 3 probabilistic models and along with the BLAST-based validation. If the combined weight score surpasses a threshold, the sequence is assigned to the corresponding phylum, or it is flagged as a new phylum. Thank you.